Dear students, and let's continue with microbial taxonomy. Previously, we had mentioned the relevance of molecular characteristics in the taxonomy of microbes, and we also did explain what is the basic of nucleic acid sequencing. And among the bacteria, as I told you, there is a region of the bacterial gene which we sequence to identify what is that bacteria. And that region, in the case of bacteria, it is a small subunit 16S ribosomal rRNA, as well as in the case of fungi, it is a 18S ribosomal rRNA. And in the case of respiring organisms, it is the part of the DNA which codes for cytochrome oxidase or the CO1 gene which we sequence to get information about that respiring organism. And if it is a plant, we take the RBCL or the Rubisco gene and we sequence it. So using this data, you can identify what particular type of organism we are characterizing. Now, when you consider the 16S ribosomal rRNA sequence in the case of bacteria, you can say that the oligonucleotide signature sequences which are mainly being found, and these are mainly being found in the 16S ribosomal rRNA regions in the case of bacteria, and either complete or more often specific ribosomal rRNA fragments can be compared in order to find out the phylogenetic relatedness between the organisms. And when you compare the ribosomal rRNA sequences between two different organisms, and their relatedness is represented by the percentage sequence homology, and you say that if they have a 70 percentage cutoff value, that is if they are being found to be related by 70 percentage, then we say that they belong to a particular species. In the previous session, we had told that by comparing the 16S ribosomal rRNA as well as the small subunitary rRNAs or the SSUR ribosomal rRNAs, we can help them to identify till the genus level. But also you can see that if the cutoff value or the similarity between the organism is about 70 percentage, then we say we can also conclude if they are of a particular species or not. Now, the next technique, the next molecular technique that which we use in taxonomy is the genomic fingerprinting technique. This is being used for the microbial classification and for the determination of phylogenetic relationships. And here you require the analysis of genes that evolve more quickly than the ribosomal rRNA. And for that, what you do is you do multi-locus sequence analysis, or the what you do is the housekeeping genes, that is the sequencing and comparison of 5 to 7 housekeeping genes is done to prevent the misleading results from analysis of one particular gene. So in this cases for the fingerprinting, rather than going for the ribosomal rRNA gene, you will try to sequence different types of genes. And when you're using, when you're trying to compare 5 to 7 housekeeping genes and then analyze it, it is called the multi-locus sequence analysis. And sometimes multi-locus sequence typing is also being done, which helps us to discriminate among the different strains. So you do keep in your mind you're sequencing different loci or the different genes and then you are comparing them. That is what is being done in the multi-locus sequence analysis as well as the multi-locus sequence typing also. So let me just give you what is an idea of fingerprinting. You got an idea that it is of course comparing different genes, but how do you do that? Let's have a look into this. What you do is you take the DNA. Okay, now if you're taking it from a human, you can take the cell or the infected tissue or whatever it is, or you can take the culture or you can take it from a plate, from a block. Now if you're taking from an infected tissue, that is if you want to identify a bacteria from an infected tissue, you can take the bacteria from the infected tissue, or you can take it from a cultural broth, or it from a agar plate. So you take and isolate the DNA, and what you do is you carry out a PCR reaction by in order to make multiple copies of that particular gene. See, you need to sequence the DNA, but you know that DNA is of small quantity. So if you want to sequence it, you need a little more amount of DNA. So what you do, you will take your DNA, and you want more DNA of your desired gene only. I told you, you will sequence only the desired gene. Now 16 is rRNA region or 18 is rRNA region. Only that particular gene is getting sequenced, or that has been getting decoded when you're doing the process. So for that, what you do, you will prepare multiple copies of that region or the gene of interest using the PCR. PCR is of DNA photocopying machine, and as a result, what happens? Large amount of your desired gene of the same is prepared. And after that, what you will do is you will carry out a electrophoresis so that the DNA will get separated. And after this, okay, uh, you will check, okay, you will check whether what they are being found to be similar or not. Okay, this is a case of genomic fingerprinting, and you will classify these organisms into small groups. Okay, the nucleic acid sequences will be obtained, and based upon that you will carry out the you will cluster them in or classify them into different organisms, and you will save them into a library. Now what is a library? That is, there is a database. You will save them into the database of the genomic database of the bacteria. Now, you might have a doubt like, how does this different from the 16SR ribosomal rRNA gene analysis? So here in genomic fingerprinting, it is not only, as I told you, it is the housekeeping genes also which will be sequenced. It is not only the 16S ribosomal rRNA that is mainly being used for barcoding and all that, which we use the 16S ribosomal RNA. In the genomic fingerprinting technique, you will try to sequence those housekeeping genes which are being present within the bacteria, so that and then you will try to identify their similarity, and then you identify. So that is a case of a fingerprinting technique in which you take the DNA, amplify the gene of interest by PCR, and after electrophoresis you will find a pattern like this, and then you will do the sequencing, and based upon the sequence analogy, the computer itself will cluster, like this is a it will prepare a what, a tree like pattern called the phylogenetic tree for you, and you can classify them into different organisms. So that's a case of you will be learning more of these bioinformatic tools in the coming semesters, so I'm not going to the details of it. And next molecular method which we have is the restriction fragment length polymorphism. Here we use restriction enzymes to recognize specific nucleotide sequences, cleavage patterns, and it is mainly by using restriction enzymes. Now, what does mean by restriction enzymes? Restriction enzymes are those enzymes which will cut DNA, okay, which will cut DNA at specific sites, and these are in fact being produced by the bacteria itself. Like you have EcoR1, BAMH1, like that, different restriction enzymes are there. And when you cut the DNA by using these restriction enzymes, they get a band pattern. And mainly by comparing this RFLP pattern, you can also identify certain bacteria. So in such cases, I'll just show you what is that. You take your DNA. No, in the case of bacteria, you take the bacterial DNA and then you cut it with different restriction enzymes. BAMH1, like that, Now here the one enzyme which we used is BAMH1. Okay, that has been cut by all the, now the isolate 1, isolate 2 and isolate 3 is the DNA is taken, and you are cutting it with a restriction enzyme. So in the isolate 1 it will form what? Two different bands. In isolate 2 it is forming only one band, and in the case of isolate 3 you find two different missing. So like that you will be able to make an implication. If two different organisms are having the same band pattern, then we can say that they are found to be more or less been found to be identical or similar to nature. But you can see that the similarity between the ribosomal rRNA genes is mainly been determined by RFLP rather than sequencing. You can also, that is called, you can use this uh, the similarity between the ribosomal rRNA genes can also be determined by RFLP also, but sequencing can also be used. But right now we use more of sequencing. That's what is more easy than going for a RFLP analysis. This is a method of RFLP, but right now we go for more sequencing strategies where the gene is being taken and sequenced. Now, the next method which we can go is the single nucleotide polymorphism. Here, what we look is, you look at single nucleotide changes or polymorphisms in specific genes. And 16S ribosomal rRNA focuses on one specific gene. And the regions targeted because they are normally conserved. So single changes in a base pair will reveal evolutionary changes. So in some bacteria, one particular nucleotide, when it is changed, there would be some changes. So we will, in single nucleotide polymorphisms, we will check whether that nucleotide base changes are also be there. So if you go to take the different types of methods which we considered over here, and their relevance in taxonomy, you can see that what? If you are doing the 16S rDNA sequencing, you can identify the bacteria up to the genus level. Okay, and if you carry out the molecular percentage of G plus C content, you can say it up to the what? Genus, species, as well as a subspecies level identification can be carried out. And if you do the multi-locus sequence typing, you can carry out, give information on the species, subspecies, as well as the strain. And the SNP analysis or the single nucleotide polymorphism will help us to identify the species, subspecies, and the strain. And the genomic fingerprinting technique will help us to identify to the species, subspecies, as well as the strain level. And if you take the whole genome of a bacteria and then sequence it, you can classify it into the family, genus, species, subspecies, and strain, and to the strain level. So using the molecular characteristics and different techniques will help us to identify the bacteria at different levels. Okay, I hope you got an idea of what are the different molecular methods which we use for the identification of bacteria as well as a bacterial species. And with this, we come to an end of the session. Thank you for now.